Hello, everyone, and welcome to the University Hospital Foundation's first ever virtual event. My name is Carolyn Thompson, and I'm so very fortunate to serve as a staff member of the University Hospital Foundation. In a moment, I will introduce you to a few of my colleagues, but first, I wanted to thank everyone for joining us. It looks like we have easily 25 people or so online. I saw the number a minute ago, and I don't see it anymore, but welcome to each and every one of you. The foundation has been very busy over these past several months as we adapt to the changes in the world and seek new ways to ensure we continue to engage with our supporters, our physicians, and our colleagues. We are so grateful for the outpouring of support to our COVID-19 Emergency Response Fund, and we continue to focus on many other funding priorities, which will advance healthcare through technology, research, and patient care. During this time, we are all likely faced with some level of challenge, and in today's environment, we are learning how to do things differently. Today's virtual brain expedition is a perfect example of that. If we can't bring you all together on site at the hospital or in a lab, we thought we'd try coming into your homes and offices. And so at least we don't have to deal with traffic, travel, or parking. In a few moments, you will hear from our President and CEO, Dr. Jody Abbott, who has the honor of introducing Dr. Jhamandas and his presentation on A Quest to Treat Alzheimer's. And towards the end of this virtual event, my colleague, Prince Osei, will facilitate a question and answer session. Behind the scenes is Elizabeth Conrad, our events coordinator, who has planned this session literally minute by minute. So what will the next 60 minutes look like? Well, you have a few more minutes with me, and then I will pass the mic over to Dr. Abbott, followed by our keynote speaker, Dr. Jhamandas. We'll finish the session off with a Q&A and then some final remarks. For everyone's reference, we are recording this session. We cannot see you or hear you, and your questions will be entered into a Q&A box. We are, of course, the University Hospital Foundation. There are many hospital and healthcare foundations across Canada, as well as right here in Edmonton. Funds raised by the University Hospital Foundation support the University of Alberta Hospital, the Mazankowski Alberta Heart Institute, and the K Edmonton Clinic. Now, did you know the University Hospital Foundation has been around for almost 50 years? The University Hospital Foundation was created in 1962 by an act in the Alberta legislature. In the decades that followed this, we have evolved into one of the most successful hospital fundraising organizations in Canada. Thanks so much to the generosity within our community and beyond. So today's presentation is, of course, on the topic of Alzheimer's, and you will sadly hear some sobering facts on this condition. Alzheimer's disease is a physical, progressive, and degenerate disease of the brain. It is one of many brain disorders. Sadly, 1 in 3 Canadians will be impacted by some type of brain disorder at some point in their lifetime, either personally affected by a brain condition or have a close family member or friend who is affected. In fact, 10% of the patients admitted into the University of Alberta Hospital are brain related. The burden of cost to the Canadian healthcare system is $22 billion annually. This equates to 14% of the total healthcare costs. So to learn a little bit more about our audience today and how you may have been impacted by brain condition, again personally or through a loved one, we invite you to participate in this live survey. The results of the survey are anonymous, and it may just show us how significant brain conditions are within this group. So here we go. If you'd like to participate and answer any of these questions.
Okay, we have some Some answers coming in. We're at 50%. All right, we'll give you guys just a few more seconds. We're at 75%. All right, so we can end the poll now and I'll share some results.
Okay, well, within this group, most predominantly is being impacted by dementia, followed by, it looks like, stroke, depression, Parkinson, brain injuries, brain tumors, MS, epilepsy. It almost goes to say within this group, the percentage may even be greater than 1 in 3. Thank you so much for participating in this. It was just, as I said, to get a bit of an idea of on our audience. And so now, it is my pleasure to introduce Dr. Jody Abbott, the President and CEO of the University Hospital Foundation, and a woman of vision who is as passionate about transforming healthcare as Dr. Jhamandas is about his research. Jody?
Thank you very much, Carolyn, and welcome everyone. Certainly when I see the survey, this also impacts me. My father is suffering from dementia, and unfortunately I lost my mother as a result of stroke. And so I think what that says is what we're going to hear from Dr. Jhamandas is very close to home, and I think it's close to home for all of us. Dr. Jack Jhamandas is a distinguished university professor from the Division of Neurology in the Department of Medicine at the University of Alberta. And he has been recognized for many of his achievements. In fact, he has received the Gold Medal in Medicine from the Royal College of Physicians and Surgeons of Canada. Dr. Jhamandas is a brilliant researcher who has dedicated much of his career to finding answers that will hopefully lead to a cure for Alzheimer's disease. Dr. Jhamandas is someone we think of when we say the impact of your support extend beyond the corridors of our hospitals and the borders of Alberta and Canada. The research you're going to hear about is truly transformational and could change the tide for tens of millions of people around the world who will be touched by, directly or indirectly, by Alzheimer's disease in the decades to come. I know that you will be in awe of Dr. Jhamandas. I know I am. Jack, thank you very much for giving us your time today, and I'd like to pass things over to you.
Let me just see if I can get this. Thank you very much, Jody, for that very kind and generous introduction. And I feel very deeply honored and privileged to have this opportunity to participate in your inaugural virtual brain expedition. And I hope that I will be able to convey a sense of interest and excitement that many of us in our group and our collaborators have been feeling for the last little while concerning Alzheimer's disease. And hope to make this an interesting and informative presentation for all of you. So, you know, this is a brain expedition, and when we think of expeditions, I'm an old Trekkie fan. So I want to just take you on a little, you know, Star Trek type expedition, because this is what it's all about. Um, I'm,
Dr. Jhamandas, sorry
Yes.
to interrupt. We just aren't seeing your screen.
oh. Are you seeing it now?
No, no.
Let me see. Can you see it now?
No, no, unfortunately not. So are you able to see the share screen button at the bottom of the?
Oh, right. Yeah. Let me just see. Yeah.
All right, here we go.
Okay. Are we on?
Yeah, so you can just make it full screen. Perfect. There you go.
Okay. So I think everybody heard what I had to say anyways. So I said that this is a brain expedition, and for someone like me who grew up in the 60s, Star Trek is the ultimate in terms of expedition. So I'm also a bit of a movie buff, and I'm going to take you on a little journey today, and I'm going to tell you a story. And the story really is that of, you know, the search for really the Holy Grail, which is treatment for Alzheimer's disease or a cure for Alzheimer's disease. And I'm going to show you a little video clip from one of my favorite movies, which is called The Indiana Jones and the Last Crusade. So I'm just going to play this movie for you. It is a clip where the actor Indiana Jones, played by Rex Harrison, goes to find this Holy Grail and eventually is able to find this you know, hall where there are all kinds of cups, but he doesn't know which one is the Holy Grail. So he picks one and takes a chance and drinks the water from this grail.
You have chosen rise. The grail cannot pass beyond the great sea. It is the bond at the price of immortality.
So I think, you know, it's not an unreasonable notion to think of, you know, a cure for Alzheimer's disease really as the Holy Grail. And, you know, this is what we are in pursuit of. So before we take this journey, let me just tell you a little bit about Alzheimer's disease and share with you some very basic facts, I think, which bring home the scope of the problem and the public health concern that we are dealing with in this time with this condition. So Alzheimer's disease is a form of dementia. It is by far the most common cause of dementia. It's estimated that there are 80 to 90% of all the dementias due to Alzheimer's disease. And dementia, in a sort of a general term, is what we refer to as global deterioration of intellectual faculties in individuals. So it's estimated that 1 in 9 at age 65 have this condition, but by 85, it's estimated that 1 in 3 individuals have this condition. And I would think that 1 in 3 may be actually an underestimate, because we are now beginning to recognize that there are conditions which are harbingers of Alzheimer's disease. So that actually it might be close to 1 in 2 almost. Now, it's estimated that, you know, in Canada, there are 500,000 people affected. And in Alberta, if you use the rule of 10, we are 1/10 of the population of Canada, 50,000 people. Now, 50,000 out of a population of Alberta, which is 4.5 million or so, maybe sometimes doesn't seem as many people, but I think you have to look at this condition through a very different lens. And what I mean by that is an individual who has Alzheimer's disease, their lives affects a lot of people around them. And as you will see in the video, you know, it affects brothers, sisters, spouses, significant others, children, grandchildren, grandparents. And so if you take 12 people being affected by an individual, you multiply 50,000 by 12, you get 600,000 people in Alberta directly or indirectly affected by this condition. So that's 1 in 7. So that is a huge sort of public health concern. And I don't have to tell you that in this day and age of healthcare costs, it's an enormous burden. And there's a figure of US $818 billion, and it is an enormous figure. And we cannot build nursing homes and facilities for our elderly fast enough, really, to deal with this. Now, I could tell you about the symptoms that people with Alzheimer's disease have, and I think many of you know that it really is many, many aspects of cognitive and intellectual function that people lose over a period of time. Now, this is a slide which I consider to be one of the most moving and evocative images of Alzheimer's disease. And this slide shows you self-portraits by an artist, Mr. Utermohlen, who lived in London, UK in the 60s. And you can see at the sort of the top right-hand part, it says 1967, and he painted himself, a very handsome young man. And, unfortunately, in 1995, less than 25 years later, he was diagnosed to have Alzheimer's disease. But he told his wife, who was also an artist, that I want to paint myself every year for as long as I can. So every year after, he tried to paint himself, and you can see 96, 97, 98, 99, and 2000 is the last year. And there is a unrelenting, progression of dissolution of self-image that is captured in these portraits, And I think, he died about 5 years after the last one, which was 2005. But his wife, Patricia, as I said, who was also an artist, said that he really died the day he could not paint anymore. So I think this conveys in a very human way what we are looking at. Now, we understand a lot more about Alzheimer's disease now than we did, 20 years ago, even 10 years ago. And I think it's a very complex condition, and I won't minimize its complexity, but we have come to appreciate that Alzheimer's disease is characterized by deposition of abnormal proteins in the brain. Okay, there are several types of proteins that are deposited in the brains of Alzheimer's patients in areas of the brain that are critical to memory, learning, and other intellectual faculties. So these proteins are considered to be toxic to the brain, okay? And two of these proteins that have attracted a lot of attention are this tau protein over here, which is found in the neurons or brain cells in areas that are important in learning and memory. And over here, you see beta amyloid plaque, or amyloid plaque as it's referred to, which contains this protein called A beta. Now, A beta is not only found in these plaques, but it's also found all over the brain in solution. And it is considered to be highly toxic to the brain. In fact, the accumulation of A beta or amyloid beta in the brain precedes the development of symptoms by years and sometimes even decades. So it's a very early event in development of Alzheimer's disease. Now, there have been a tremendous amount of efforts made to say if amyloid is so toxic to the brain and produces dementia, can we somehow prevent its generation in the brain or get rid of it somehow? And really, that's what the focus of this field has been for the last 20 years. And, as I said on the slide, what have we done about this impending tsunami? And I think this is actually a very depressing slide, I'm afraid, and I apologize for showing it in some ways. But what it tells you is that there's been no shortage of effort. There have been 413 clinical trials done testing 244 compounds to find a way to fundamentally alter the course of this condition. And unfortunately, all of these efforts have failed to produce a single drug. So this is the despair part of the story that I am going to tell you about. Well, you know, about 20 years ago, we developed a notion in our laboratory that, you know, there were certain drugs that had been developed for diabetes, and we had perhaps the notion that maybe some of these drugs might be beneficial also in treating Alzheimer's disease. Now, the sidebar to this conversation is that there is a very interesting link between diabetes and Alzheimer's disease. People have, for quite some time, appreciated the notion that if you have diabetes, your chances of developing Alzheimer's disease are significantly higher than if you don't have this condition. So, you know, there is this, what we call epidemiological link between these two very important and common conditions. So in the 1990s, people had developed certain drugs to treat diabetes. And one of those drugs is a compound referred to as AC253. And we began to do studies on this drug AC253 to see if it could protect brain cells in the dish from the toxic effects of amyloid. Now, for those of you who are at this point wondering, have I heard of AC253 before? I can assure you, you have, because there is a flight from Edmonton to Vancouver, which is AC253, leaves every day and goes to Vancouver. You know, I like to think that the AC253 compound we are working on has a better odds of getting us where we want to go than this flight, where you are likely to not arrive on time, probably lose your luggage, and or maybe altogether not get there because they have canceled your flight or something like that. So anyways, AC253 is the drug that we have been working on. Now, in the 1990s, there was a tremendous advance in the field of Alzheimer's disease. And that advance came from the fact that, you know, to test drugs in Alzheimer's disease, you need animal models to be able to test them before you can put them in human beings. And in the mid-90s, people were able to genetically engineer mice to develop Alzheimer's disease. And this was a phenomenal advance for the field. Now, I won't go into the details of how they did that, but this slide shows you a normal brain of a mouse. This is the back of the brain. This is the front of the brain. This is the area called the cortex, and this is the area called the hippocampus, which is the seat of learning and memory. Now, below here, you see a similar section of the brain, but this section is from the brain of an animal that has developed, a mouse that has developed Alzheimer's disease. And each of these green dots that you see on your screen on this section represent amyloid plaques. And you know, you don't have to be a neuroscientist to see that there is a clear difference between these two brains. This brain from the Alzheimer mouse is just riddled with plaques, much like a human brain would be developed. So this model isn't perfect, but it has been a major advance in terms of testing our ability to see if compounds that, you know, do work in Alzheimer's disease, or maybe they could help. So we have injected this compound, AC253, by injection and directly into the brain of animals and tested their memory and learning. And obviously, the question is, how do you test memory and learning in a mouse? And you know, there are many, many ways to do this, but one of the tests we use is something we call the Morris water maze. So this is a pool, a 6-foot diameter pool of water, and this white disc here is a platform, okay? So what we do is we put the mouse in this pool, and this red arrow shows you the path of the mouse. He or she wants to find this platform, and we can time them. So you can see that this guy made a beeline. He was able to get on the platform. This is a normal mouse. Now, the first day, it will probably take him around 60 to 70 seconds to find this platform. But every day, he's going to get, she or she is going to get better and learn the task. When they learn the task, by day 7, they can do it in 10 seconds, okay? Now, you know, it's not unlike human situation, where imagine you have moved to a new neighborhood and your grocery store is about 15 minutes away. Now, the first day, it's going to take you 15 minutes to get to the grocery store, maybe even longer. But subsequent days, you're going to get there quicker because now you have used cues in your environment to know where the grocery store is, and you're going to take some shortcuts to get there. So by the end of the week, you'll probably do it in 10 minutes, okay? So this task is not dissimilar to that type of a thing. Now, in Alzheimer's mice, which is shown in this slide, if you put the Alzheimer mice in the pool, look at what this mouse does. I can't tell you whether it's a he or a she, but we have speeded this up just to show you that they spend a lot of time meandering around in the pool to find this platform. Now, you know, eventually they'll find it, but it's going to take them a lot longer than a mouse of the same age who doesn't have Alzheimer's disease. Now, these guys will also get better if you test them every day because they'll learn where the platform is. But they will never get as good as the normal mice. So here is some data that I want to show you. And this red trace and the blue trace at the bottom here represent the time it takes for the normal aged mice to find the platform. Now, the first day, it's about 60 to 80, 75 seconds to find it. But by day 7, they can find it in 10 seconds or so, okay? Now, both of these sets of mice are normal mice that got our drug AC253. And of course, you know, you don't see any effect of the drug on normal mice. In green over here, you see the time it takes for the Alzheimer mice to get to the platform. Now, they also learn on subsequent days, but they never get as good as these guys who don't have Alzheimer's disease. Now, in purple over here is traces from groups of mice that got this drug AC253. And you can see that these guys improve their memory and learning, so they're a lot closer to the normal non-Alzheimer's mice than they are to the ones that did not get the drug. So this got us very excited about, you know, the potential of a drug like AC253 to improve memory and learning. Now, the problem with AC253 is that it is what we call a peptide. Now, peptide is a type of a molecule that consists of a string of amino acids, okay? So AC253 is 25 amino acids strung together. Now, unfortunately, peptides, when you take them by mouth or even inject them, get metabolized in the blood because there are enzymes that chew up the drug. And so not enough of it can get across the brain. And because of their size, they have problems getting across the brain anyways. And you know, when you put amino acids in the body, there is always a risk that you're going to set off some type of an immune reaction in the brain. And in fact, that was one of the things that resulted in vaccines not being a viable therapy so far, because they would set off essentially an immunological bomb in the brain. And the other advantage of something we call small molecules is that you can take them by mouth. Small molecules are drugs that you take for your blood pressure, for your cholesterol, for your heartburn and things like that. And of course, pharmaceutical companies are very interested in small molecules because they're a lot cheaper to make than there are peptides. So, you know, about a year and a half ago, I hooked up with Dr. Lauren Terrell, who's a famous virologist here. Lauren discovered the first treatment for hepatitis B right here at the University of Alberta. And my other collaborator was Dr. Michael Houghton, who's the discoverer of the Hep C virus. And these two individuals are leaders in the field of virology. In fact, they are right now working on COVID vaccines and trying to develop drugs for COVID. But these two individuals set up the Virology Institute. And the Virology Institute has many strengths, but the ones that we were particularly interested was that they had the ability to model, computer model drugs and use artificial intelligence to identify small molecules that might be important. The other advantage they have is that they have a group of chemists who can make drugs for you if you tell them what the structure of the drug is. So these were the two things that were very important for our voyage. And this slide shows you what computer modeling and artificial intelligence is all about. Now, these ribbons, this purple ribbon here and the red ribbon here, is something we call the amylin receptor. So the simplest way to think of a receptor is a keyhole, okay? It's like every door has a keyhole. Now, the peptides like AC253 are keys that fit into those keyholes, or they can block that keyhole. And what Lauren's group and Michael's group, who was a very talented computer modeler by the name of Kamlesh Sahu, what he did was he modeled these receptors or keyholes, and he took our data on AC253 to see where the key would fit into the keyhole, and try to find if there were small molecules that we could find that would be promising and potentially treatments for Alzheimer's disease. So with this approach, we were able to screen 7 million molecules using a computer called BlueGene, which resides in Southern Ontario, but the virology department here, Virology Institute, has some shares in, so that they can use it. And 7 million compounds. This took us 3 weeks. In the old days, it would have taken us 5 years to do this. And out of that, we were able to find one compound, this 9012, that seemed to hold the promise and potential much like AC253. So we can take 9012, which is this compound over here, and apply it to brain cells to see if it will protect them from amyloid. So brain cells can be grown in a dish over here, and you can see them, they're all green here. So what you got to remember on the slide is green is alive and red is dead. When you put amyloid on these brain cells, and these are human brain cells, you can see that amyloid kills these cells. So there's a lot of red over here. This is just amyloid, no 9012 or the compound right now. As you put increasing doses of 9012 right over here to the right, you can see that there are very few red dots here. So the point is that this drug could protect nerve cells in the dish, which got us very, very excited. So the next thing was, can we restore memory using these compounds? So for that, we use two approaches. One is, believe it or not, you can actually record memory in a dish from the brain of a mouse. What you do is you take a piece of the mouse brain, which contains an area we call the hippocampus, which is the seat of all learning and memory in humans and in mice. And if you take this brain slice, you can keep it alive for hours on end if you perfuse it or if you apply, artificial spinal fluid on it. So you can keep these brain slices alive for hours on end. And what you can do is if you apply shocks to this brain slice, what you get is you get a huge burst of increase in the signal. And the signal that was here goes from here to here, and it can last for hours on end. And we call this long-term potentiation or the memory trace, okay? And it's a very important sort of correlate of memory in living human beings. So it turns out that if you look at this memory in a dish, in Alzheimer's mice, and I've already told you about these mice that have a lot of amyloid in the brain, you can see, focus on this blue trace over here. The memory trace is very, very low, compared to the trace, which you can't see quite that well over here, which is in the black, which is from normal mice. But if you put 9012, this compound we had synthesized on the brain slices from these Alzheimer mice, you can restore this memory trace in blue here to a level which is shown by yellow here, which is very close to what you see in normal mice. So this got us very, very excited that not only could this drug protect nerve cells, but it could also help memory and learning. Now, we also, as I've shown you previously, test memory and learning in this big pool, and these mice swim around, and their job is to find this platform. We can time them. We can say, look, you know, first day it's going to take this guy, it's the first day he's done it, 70, 80 seconds, but by day 5 or day 7, he or she is going to get much better. So what we did was we took these mice and we took Alzheimer's mice of the same age. Now these are mice that had Alzheimer's disease. And we gave them 9012, this compound that we had generated by injection every day for 5 weeks. And here is the data that we saw. The green traces over here are normal mice. And as before, you can see these guys get better. The Alzheimer's mice over here in red don't get better. They haven't got the drug. But the red trace over here are the mice that got 9012. And they are a lot closer to here than they are to there. So what this told us is that not only could it restore memory in the dish, but it was also capable of restoring memory in mice that already had Alzheimer's disease. One of the things we also looked at in these mice was to look at their brain pathology. Did it do anything to amyloid burden that these mice have, you know, or at least the Alzheimer's mice have? So this is an Alzheimer mouse, and you can see each of these blue dots over here is an amyloid plaque, lots of it. This is the brain section, similar part of the brain, from a mouse that got the drug 9012. And you can clearly see that there is a reduction of the amyloid burden and plaques here compared to here. Now, one of the other things that happens in Alzheimer's disease is that there's a lot of inflammation in the brain. And this is what inflammation looks like in the brain. Each of these green things is what we call microglia, which are these cells that are involved in inflammation. And you can see them at higher power here. They look these sort of angry looking cells. This is in a normal Alzheimer mouse, top and bottom on the left here. Here is a mouse that got the drug. And I think you can clearly see that there is significantly less inflammation here than there is here. Now, you know, we are now really moving this program forward at a fairly rapid rate. We have shown that 9012 can improve learning and memory. Not only that, it can also reverse some of the brain changes that we see in Alzheimer's disease. Now, one of the things with 9012 is that we have to give it by injection. And really the holy grail is to be able to give the drug by mouth. So one of the things that we have been working on, and we are almost ready to start these experiments, is that we now have drugs that are based on 9012 that can get into the brain much more easily when we give it by mouth. And so oral formulations, we are going to put these in mice. We are going to see their memory and learning over time. And once we see benefits of that, then we can start to move this into clinical trials. Now, you know, the last thing here is that research is an expensive business. And, you know, one has to raise funding to be able to continue to develop and test efficacy of these drugs. And, you know, we have been fortunate to have funding from the University Hospital Foundation and yourselves as donors that has allowed us to be able to do this. I want to just close off by acknowledging people. You know, these are people in my laboratory and the team from Dr. Terrell and Houghton's group over here. These individuals have been critical in every facet of the story that I've told you. We have got funding from national and provincial agencies, but we have had tremendous support from the University Hospital Foundation, because this is really what I call high risk, high reward type of science, which many of the times, agencies like Canadian Institutes of Health Research and stuff are a little uneasy about funding. But, that's what it takes sometimes to go where no one has been before. And I'm just going to leave you with this slide, which is from a place in Africa close to where I was born. And the people who live on this crater, the Ngorongoro crater, they don't have a lot of food, they don't have water. They're lucky if they have a shelter. But, every day they get up in the morning and they have hope that that day will be a better day than the one they've had. And I would like to leave you with the notion that, we have hope that, our Alzheimer's patients will have a better tomorrow than we have today. Thank you very much.
Well, thank you so much, Dr. Jhamandas. I'm going to let my colleagues formally thank you in a moment, but first I just want to set up a video. So typically on our brain expeditions, we're able to take some donors into the laboratory to go on a tour. And of course, that's not really a possibility right now. So we wanted to be able to share Dr. Jhamandas' lab with you all.
Alzheimer's disease is a form of dementia. It's estimated that there are about 500,000 people in Canada that have Alzheimer's disease. Put it in an Alberta context, that would be about 50,000 Albertans. Their life affects, at the minimum, 12 people around them. Would be their spouse, would be their significant other, parents, uncles, brothers, sisters, children, grandchildren. So 12 is not an unreasonable figure of people that are affected by a particular person. So multiply 50,000 by 12 and you get 600,000. So that's 1 in 7 Albertans who are directly or indirectly affected by this condition. The incidence of this condition is going to escalate very rapidly, so that in the next decade, it's estimated that we may have 50 million people who have this particular condition. We have been working in the last few years on trying to develop drugs that would protect brain cells that are affected in Alzheimer's disease. So we have identified a group of compounds that we feel hold promise and potential for protecting those brain cells. We have found that in animal models of Alzheimer's disease, that is, these are mice that develop Alzheimer's disease, administration of these drugs can actually improve learning and memory in this. So there is a tremendous sense of excitement that we are on the right track to find something that will fundamentally alter the course of this disease. So what we do is we fill up a pool, about a 6-foot pool of water, and it's got this platform in here. And what we do is we have a mouse released into this pool. And this guy is going to scoot around and he's going to try and locate this platform. Now this is his first day in the pool, so he's going to have some learning to do to find out where exactly this platform is. And so you'll see he'll jump on the platform and we can then say cut, time it. Now this is mice that have got the drug for a period of time, and these 2 traces at the bottom are mice that are non-Alzheimer mice. So these are normal mice, and you can see they start around 60-70 seconds to find the platform, but by day 7 they are down to around 10 seconds. They know it. The guys in green over here are Alzheimer's mice, and they too learn, but they're never as good as these guys. The guys in purple here are the mice that got the drug, but you can clearly see that these guys in purple are better than those guys. So this is the type of testing we do to find efficacy of drugs for Alzheimer's. We did not see any side effects of nature that were disturbing to us. So now we are really on the threshold of sort of embarking upon another sort of journey on this, which is to test drugs that we can be administered by mouth that get in sufficient quantities in the brain and produce the same beneficial effects that we saw with injections of these compounds. This research is the type of research that requires philanthropic support. This allows us to be able to continue with that research on an unrelenting basis. I would say without any hesitation that the donors, particularly the University Hospital Foundation donors, have played a critical role in helping us get to where we are. One of the key aspects of research is what we call sustainability. You have a good idea and you develop that idea, you get results, and then all of a sudden you don't have any more money to take it to the next step. Time is money in research, and we need that support to get us to the finish line. I would like to say a big thank you, first of all, and an expression of gratitude that comes from the heart for all the things that you have done. It doesn't matter how much you have donated. The fact that you became a partner in this and you became a supporter in this is immeasurable. The second thing I want to tell you is that don't lose hope. We have a real sense of excitement right now, and I think it will be of great interest to patients and families who have this condition. Hope is something that we need a lot of these days, and particularly in conditions like Alzheimer's, where we have really hit a lot of brick walls, so to speak. I'd like to leave you with the notion that what we are doing should be a source of hope for the future, hope that can bring life.
Prince, you're still on mute.
Thank you so much, Dr. Jhamandas, for taking us on this exciting and educative expedition, and for also giving us a tour of your lab. We will now jump into the Q&A. So for the audience, if you look at the bottom of your screen, you should see a Q&A with a sign. There's a dialog box in there. We've got 10 to 12 minutes allocated for our Q&A. Please type in your questions. I will read the questions to Dr. Jhamandas, and then he will respond live for everyone to see. If by any chance we do not get to all the questions, we will try to get the answers and we will send them to you later. So we will jump right into it. I see there's a first question. So the question is, there are several drugs which have been used over the years to help Alzheimer's patients. Were any of these drugs part of the study of the chemicals that you evaluated in your computer simulation?
So, currently for Alzheimer's disease, really, we have no drug that can fundamentally alter the course of this condition. We have drugs that can treat the symptoms, and they do so to a point and for a period of time, but then they lose their efficacy because the degeneration of brain cells is relentless and continues. Now, with respect to our sort of modeling and stuff, we modeled these drugs based on the receptor or the keyhole that we have been working on. So none of the drugs, the usual sort of suspects, so to speak, fell out of the computer modeling. I will tell you that we screened 7 million compounds, but we put a lot of stringent conditions on identification of drugs that fulfill certain criteria. So the first pass, we got 100 some drugs, and then we whittled those down to 10. And then of those 10, we found a couple that our chemists had to actually make in-house because they were not available on shelf of any company that was there. So, you know, a lot of this was done in-house. And, you know, I'm sure the drugs that you talked about were amongst the 244 drugs that I showed on the slide that were put through 413 some trials and, you know, didn't yield anything really.
Thank you, Dr. Jhamandas. So there was a question that had been sent in, and it's a reflection of the times that we find ourselves in. Does COVID-19 render patients with Alzheimer's more vulnerable?
So this is actually a very, very interesting and important question, I think, because I think we know that COVID has affected what we call vulnerable populations. And the elderly and the aged are one group of these vulnerable population. And obviously, Alzheimer's is in the main an age-dependent condition. So as you get older, you accumulate a greater risk for Alzheimer's disease. But even above and beyond age itself, there is a sort of a thinking right now that, you know, the inflammation that is caused by COVID in the body, you know, the brain is not absolved of that inflammation. And you know, I've already told you that Alzheimer's disease, there are some changes in inflammation in the brain. So think of COVID as being a double whammy, if you wish, for, you know, you got inflammation from Alzheimer's, which is compounded by inflammation from COVID. That doesn't happen in everybody, but it does render Alzheimer's patients potentially more susceptible to COVID and effects of COVID.
Thank you. In relation to COVID, is the next question we have is that is the work on is the work on the is COVID slowing down the work on Alzheimer's?
Well, you know, COVID has slowed down all of our lives, I would say. You know, when this happened, when COVID, the sort of, you know, the worldwide sort of pandemic was declared in March, obviously university was shut down really, you know, all aspects, teaching, research, really no one was around. And so for a few weeks, we could not even step into our laboratory, but we were able to get a special dispensation from the university and our faculty to keep some of the experiments that we had going, because, you know, we had mice that were in certain critical, you know, periods of aging and developing Alzheimer's disease. And had we not been able to work on them, we would have lost a huge sort of resource and colonies, you know, to test these drugs. So, you know, we were gradually able to sort of keep the home fires burning, so to speak. And now I like to think that we are ramping up and, you know, are really at coming up to the speed that we were at.
Thank you. There was a question that had also been sent in. Is physical exercise or mental stimulation useful in preventing dementia?
Well, you know, there has been a notion and a debate that, you know, physical exercise and mental stimulation or intellectual stimulation do help, you know, ward off, if you wish, the onset of dementia. And I think that, you know, there are several things in play here. Obviously, you know, patients who have Alzheimer's disease are, you know, not absolved from having other conditions like heart disease or high blood pressure or many things. And physical exercise is beneficial for cardiovascular health and many other types of health. But I think there's a growing body of evidence that both physical exercise and mental stimulation do play a role in perhaps delaying the onset of dementia, or certainly helping when it comes on, the symptoms may not be as severe. So I think that's a field that's still in progress, but I think the available evidence seems to be pointing to a positive way.
And I'm curious to know, is Alzheimer's hereditary disease?
You know, Alzheimer's disease in the main is not really what I would call a hereditary or familial condition. It's estimated that, you know, between 5 and 10% of all Alzheimer's cases have a familial or a genetic origin. And there are specific genes that have been linked to the development of Alzheimer's disease. Now, you know, interestingly, if in those families where these genes are present, when patients develop Alzheimer's disease, it tends to be earlier onset than the garden variety Alzheimer's disease, which is a more later onset in the 60s, 70s. So patients may develop dementia, you know, due to a genetic cause in their 40s even and 50s. And when it occurs, it tends to be a bit more severe than the other forms of Alzheimer's disease. And while it's uncommon, the genetic form, it has taught us a lot about, you know, what do these genes do? What do they, and many of these genes have been identified to promote development of amyloid in the brain, neurofibrillary tangles, this other type of abnormal protein in the brain. So, you know, we have learned a lot about the biology of this condition from the small percentage of cases that we can now think about and apply to the larger cases that we see, you know, which are not genetic.
And are there any differences between the onset of Alzheimer's between women and men, the sexes?
No, Alzheimer's disease is more common in women than in men. You know, part of that is maybe women live longer, the life expectancy is a bit more than, but there may be other factors as well. You know, we don't quite have a fix on that, but you know, but when it does occur, the profile of the condition is not dissimilar between males and females.
So there is another question from our audience, and does Alzheimer's appear more often in people with other health conditions? So we're looking like, for example, MS, Lyme disease, lupus, or head injuries?
No, I don't think there's any clear-cut evidence from epidemiological or other studies that, you know, it's more common in MS or in these other autoimmune conditions that are listed on by Prince. But I did mention and that there is a clear, you know, link with diabetes and Alzheimer's disease, and you know, diabetes also being an autoimmune condition. Now, interestingly, you know, I'm just going to try and bring this back to COVID. Diabetes, you know, COVID affects diabetics, you know, much more than it affects non-diabetics. So, you know, now you can start to see the sort of connecting the dots, you know, between diabetes, Alzheimer's disease, and potentially even COVID, you know, so there may be some relationship there. But, you know, in response to the question, no, there's no clear evidence that it's more common in MS or in lupus or things like that. Head injury, you know, there is this chronic traumatic encephalopathy, which is a, you know, condition that we see in people who play contact sports, boxing, you know, football and stuff, but that's a bit different entity than Alzheimer's disease.
We've got a minute and I will take the liberty to ask my question. Dr. Jhamandas, what keeps you awake at night? What keeps you awake at night?
Well, you know, a lot of things keep you awake at night. These days, it's mostly what are we going to do with COVID-19, you know. But no, I think that, you know, what keeps me at night is that, you know, I think, you know, we are making good progress. I think it's important to think and rethink about the type of things you do and not lose sight of, you know, the sort of the end goal or the finish line. And, you know, it's very difficult right now in this environment to be able to be doing research and, even fundraising and many things that are all sort of part of the same equation. The economics of the country and the economics worldwide, research is not absolved from the impact of those things. So you worry about a lot of that. Because we have COVID doesn't mean that all the other conditions will disappear. They may be on the back burners for now, but they're very much in play. And I think the thing is not to lose sight of those.
Thank you so much, Dr. Jhamandas. I want to thank our audience for joining us today. And for the questions that came in to make it a very lively expedition. Nothing is more important than for us to hear from you. And so within the next couple of days, you'll be hearing from us. We'll send you a short survey so that you can let us know how we did and what other areas in brain care or other areas of care that you would want us to host on such a platform. And then we will also be including a link to today's recorded expedition so that you will have it on your desktop or in your inbox and be able to refer to it. Thank you so much for joining us today.
And once again, on behalf of Jody, Prince, Elizabeth, myself, and of course, Dr. Jhamandas, thank you for your time and your active participation. We hope you found today's session informative and are inspired by what the future may hold. If you'd like to learn more about how you can support the University Hospital Foundation and Dr. Jhamandas' research in finding a cure for Alzheimer's, please do reach out to us. Thank you all for helping us make history, the University Hospital Foundation's first virtual event. And I leave you now with a short video entitled A Beautiful Mind. We look forward to seeing you again soon.
Your brain never rests. Your brain never rests. It guides your consciousness, imagination, perception, memory, judgment, and emotions. From the brilliance of Mozart's symphonies to the simple act of sipping a coffee, the brain controls it all. So when something goes wrong, the results can be devastating. This is when the struggle to survive begins. Thanks to your support, the best and brightest medical minds are working together to create something amazing, like the most advanced brain surgery suites in the country, breakthroughs in stroke care, including Canada's first stroke ambulance. At the University of Alberta Hospital's Brain Centre, we are tackling the most serious of brain illnesses, developing world-leading research in MS, Parkinson's, and Alzheimer's. But it took more than brain power to get us here. Without the leadership, dedication, and generosity from people just like you and thousands of others, we wouldn't be where we are today. The University Hospital Foundation's Brain Centre has already changed and saved thousands of lives. But there's still so much more to do. With your support, we can continue to push the boundaries of care and knowledge and make sure the University of Alberta Hospital remains a world leader in brain care.